have to say anything. We've already had this incredible introduction to our next speaker, Hilary Calvert from University College London. Take it away.
Well, thank you. I'd like to thank Michigan State University and Jim Hesley for inviting me to come here. It's actually the first time that I've been here. I'll try to live up to the workup I had from Mike. I first got into oncology as a junior doctor when you only had a 6-month job at a time and you had to apply for another one. And I needed a job and I applied for the heart hospital, the lung hospital, and the cancer hospital. And the cancer hospital gave me the first offer. And when I got there, I found my boss was a quite small but quite scary woman called Eve Wiltshire. And she said, "The boys in the lab have given us some of this platinum stuff. Why don't you give it to Mrs. Smith?" So I did. And Mrs. Smith promptly vomited her heart out for several days and went into kidney failure. Fortunately, the previous job I'd done had been in nephrology, so I knew how to rescue her kidneys. And once she'd got better, we suddenly noticed she didn't have cancer anymore. And I thought, oh, maybe there's something in this oncology lark after all. So that's how it started. So the first drug I got to play with when I was the boss was Carboplatin, and you've heard about how that happened. My conflicts, I do consult for one company still, but I've never had any personal income from platinum drugs of any kind. What I'm going to cover is the early clinical development, various difficulties we had to get through, getting the right dose, which still is an issue, and then I'll look at some of the individual types of cancer and go through what the role of carboplatin is in them. So I think I can see this picture many times, and we can move straight through. In fact, this is the doctor's version of the synthesis of carboplatin, and Fuller's extra special bitter is something like Sam Adams or something like that. But I did feel that these chemists, sometimes don't make it clear how you make things. So at the time, the Royal Marsden Hospital didn't have much of a track record for doing phase 1 or experimental drug studies. It more specialized in giving state-of-the-art but existing treatments to patients who were there. And there was a regulatory framework that I was able to use, which was called a doctors and dentists exemption. And if you were going to do this, you wrote to the regulatory body and you said, I've got this compound, I can prove what it is, and I'm going to give it to people, and I take all the responsibility for what happens. If you could do that, you didn't have to submit any data or anything else, but it was entirely my fault if anything went wrong. Now, of course, I did have data. We had all the studies that Mike's been talking about, and we knew from the preclinical animal studies that it was likely to be pretty safe. So this did allow us to get into the clinic very quickly. And this personal exemption on me actually took the drug all the way through to the end of phase 3 before Bristol Myers took an interest and set up trials in a more formal way. So when we did a phase 1 study, we escalated the doses up to 520 milligrams per square meter. We had very detailed monitoring of renal function using isotope tests, and that showed no significant changes. We did audiography on everybody because deafness can be a problem with cisplatin, and showed no deficit. Neuropathy, it can be a problem with cisplatin. We didn't see any new neuropathy in the patients, but some who'd had cisplatin before it got a bit worse. The dose-limiting toxicity was the platelets, thrombocytopenia, which is quite unusual. And although leukopenia was seen, it was only about the same as what you see with cisplatin. There was nausea and vomiting. It's hard to quantify, but the feeling was it wasn't really as bad as with cisplatin. But the other thing that happened in this phase 1 study is we saw quite a lot of clinical responses. And a complete response is when the tumours all disappeared. A partial response means it's got to about 30% of its original size. And you can see, particularly in ovarian cancer, we did see quite a number of responses. And again, this is really an unusual finding in a phase 1 trial. So we seem to have very low non-hematological toxicities and good signs of activity. And I was expecting everybody to sort of cheer and we could go ahead from there. But like many things, it didn't quite happen that way. The first was I was the new boy. I'd only just been made a consultant, and these are some of the more traditional consultants at the Royal Marsden Hospital at the time. And they didn't like it, and they thought I was taking too many resources that they could maybe use for their private practice or something. And they managed to get the hospital to ban me from admitting patients. Eventually, I got it turned around, and I was able to carry on. At the first presentation we gave to the early clinical trials group in Brussels, it got really quite a hostile reception. I think they didn't know me. It was the first time I presented a phase 1 trial, and they'd done lots. And they just really didn't believe it. And even at the early platinum meetings, much more of the interest was in the hydration and diuretic treatment for cisplatin nephrotoxicity, which was felt to have overcome the problem, so you didn't really need carboplatin. The other sort of thing in the offing was that Bristol Myers had licensed a drug called spiroplatin with a sulfate ligand, which in preclinical systems showed non-cross resistance to cisplatin, which of course carboplatin didn't. And they'd had reported a response to breast cancer in phase 1, and this was Bristol Myers' favored candidate. So, in any case, Johnson Matthey, as Mike just said, set up a GM plant to make carboplatin, and Bristol Myers offered to formulate it for us. The first batch that went formulation got lost, which put us in a bit of a pickle because we had patients lined up for the trials and no drug. Fortunately, at that time, the NCI were beginning to think they would do their own trials, so they had stocks of formulated material, although they hadn't started trials. And I got in touch with them, and Dr. Dan Hote was very helpful and released a substantial amount of formulated carboplatin for me to carry on. Eventually, spiroplatin was taken out of development because it didn't work, and it gave everybody a nephrotic syndrome. So that really put carboplatin at the forefront. So Eve Wiltshire, my previous boss and colleague, had a large ovarian cancer practice, and she started doing a randomized trial comparing 100 milligrams per square meter of cisplatin with 400 milligrams per square meter of carboplatin. And again, we did very accurate measurements of toxicity. And you can see here the complete and partial remissions were very similar for both arms. Nephrotoxicity, 71% for cis and 12% for carbo. Ototoxicity, none at all for carbo, 65% for cis. Peripheral neuropathy, a bit for cis, none for carbo. In fact, all the toxicities except for thrombocytopenia favored carboplatin very strongly. This is a picture you've seen before, which led after the launch of carboplatin. Even so, the medical community at large wasn't that keen, and Dan von Hoff, who's very famous and in fact an oncologist I respect a lot in the USA, wrote a leading article in the Journal of Clinical Oncology essentially saying this development was a waste of time and money, and we should have spent our time and money trying to make non-cross resistant drugs. Now, one of the things about carboplatin when we did the phase 1 study was that the toxicities were, thrombocytopenia was hard to predict. So on this chart, these are dose levels, and the spot is how low the platelets went, and you really don't want them going much below 50 on this level. And you can see that one patient at 200 milligrams per square meter got low platelets, and 3 patients at 520 didn't get low platelets. So there's inconsistency about what happens to the platelets. And I was taken with this and wondered if we could find an explanation based on the pharmacokinetics of carboplatin. Now, at the time, people were beginning to talk about systemic exposure of cancer drugs. That is, the concentration times the time, which of course is a bit like the law of mass action for chemists. And they were beginning to notice that the toxicity of a drug was more closely related to the concentration times time, or area under the curve, than the actual dose given. And in fact, the concentration times time would cross between species quite well, while the actual dose wouldn't. And I reasoned that the cell kill to the bone marrow or the tumor would be proportional to the number of adducts formed, and the number of adducts we knew were formed what was proportional concentration times time. So we looked to see if we could predict the AUC for carboplatin. Carboplatin's got quite different pharmacokinetics to those of cisplatin. It's got a longer plasma half-life. Cisplatin reacts with plasma proteins quite rapidly, and only about 20 or 30% of the dose excreted in the urine. While carboplatin is relatively stable, greater than 80% of the dose excreted in the urine, and it's excreted at a rate that's closely related to the glomerular filtration rate. And these are data from the phase 1 study, and they show the pharmacokinetic profile of one patient receiving carboplatin. The top line is the total platinum. The lower two lines are actual carboplatin measured by HPLC or ultrafiltrable platinum, and they're almost on top of each other. So the difference between the two is due to protein binding of the carboplatin. And all the studies we did actually used ultrafiltrable platinum because it was easier to do than to measure the intact drug. Here's the amount of urine excreted in 24 hours, and you can see in 24 hours it comes up to 65%. And if you went on for 48 hours, you got a bit more. And here's the correlation between the glomerular filtration rate of the patient's kidneys and the carboplatin clearance. And you can see that it's a really good correlation. You could almost use carboplatin for measuring the glomerular filtration rate, I think. So, putting those together, comparing the glomerular filtration rate, which was measured at 51 chromium EDTA, to the total plasma clearance, we found we got a good correlation with an intersect just above the origin. And this led to us deriving a formula to say what the area under the curve would be with a constant plus 1.2 times the glomerular filtration rate. And this was a provisional formula we were able to use to see if the system actually worked for getting a predictable AUC in patients. This is the pharmacokinetic model used. Many people would say that's too simple, that you have two compartments at least for distribution of the drug. Well, indeed, we did solve a two-compartment model as well, but the actual numbers that came out of it at the end of the day were hardly different from using a one-compartment model. So we stuck with the one-compartment model because it made a much simpler formula. So after we did a prospective study, we found the formula worked pretty well, but there was some bias in it, and we came up with the final formula, which is shown here. And this is the one that's become dubbed the Calvert formula. I didn't call it that, other people did. And it's used universally for calculating the dose of carboplatin. Now, other people have developed formulas. Meryl Egerin developed one around about the same time as us, and Etienne Chatelou developed one later, which was based on the plasma creatinine rather than having to measure the 51 chromium EDTA clearance. Now, to show you what sort of effect dosing by formula has, these are 3 real patients who I treated in Newcastle. The top one was a young man with testicular teratoma, and he had a very high clearance, 177 mLs per minute, so he got more than a gram of carboplatin. He landed up in an AUC of 5.2. Our target was 5, so that wasn't bad. There's somebody in the middle, a clearance of about 100, got 600 milligrams, and landed up with an AUC of 4.3, a bit under. And this patient here had renal failure and had 5 mLs per minute clearance. Was dosed with 125 milligrams and had a AUC of just under 4. So here we have 3 patients getting very similar AUCs within 20% of each other, but there's an 8-fold difference in the dose needed to achieve that, which really helps to show why formula-based dosing is so necessary. Now, formulas do make money, and this is, I can't remember where I found that, but this is Michigan State's royalties. And the carboplatin are the blue arms, and you can see that when my final formula finally came in, the royalties really began to take off. Now, this wasn't due to chance. I was contacted in about 1989 by a man called Frank Pascalone, who was product manager for carboplatin in the USA. And he'd found it hard to get physicians to prescribe carboplatin because they'd try a few patients and then somebody get bad thrombocytopenia and they say, oh, this stuff is too difficult to handle, we'll go back to cisplatin. So he persuaded everybody to dose using my formula, and then it became the standard drug because the advantages of the reduced non-hematological toxicities and everything could be exploited. So we'll talk about some of the individual cancers. In ovarian cancer, carboplatin has taken over as the treatment of choice. This was the result of Eve Wiltshaw's first randomized trial showing overall survival being really identical. This is probably the study that really consolidated the position for carboplatin, and this was done in combination with Paclitaxel. And if you look at the top two curves, they're patients who are at a good risk with minimal residual disease who were treated with the combination. And the dotted line is the carboplatin, and so I don't think the statistics are significant, but it's doing slightly better in the long term than the cis. Then these are the patients who are at a worse risk, and again, the cis and carbo are doing almost exactly the same for survival. And that was a big trial with lots of patients, and I think has really confirmed what's going on. Now, one thing about ovarian cancer is that it's a relapsing disease. You'll get a response in most patients, but sometimes after 6 months, sometimes after a year, sometimes after 4 years, they'll relapse. And it's been known that if they relapse after a long period, they're likely to respond to re-challenge with a platinum agent. If it's a short period, they won't. Now, with carboplatin, you can re-challenge, but with cisplatin, it's more difficult because every time you give another course, you lose a bit more renal function. It's going to get to the point where you can't give it again. And this has resulted in something that I think we hadn't really come across before, is that quite often with these relapsing and re-challenged patients, eventually they'll develop a cisplatin sensitivity. They'll come out in a rash or even get something approaching anaphylaxis. And that can be a problem for repeated treatment. Sometimes it's possible to desensitize them. The other thing that really helped carboplatin along, especially for ovarian cancer, was the combination with Taxol. Because if you give carboplatin and Taxol, not only does it work well as a combination in the responses in ovarian cancer, but it also seems that you get less thrombocytopenia than you do if you give carboplatin on its own. And on this graph, which was done by Meryl Egerin, the curve on the right here shows the increasing amount of thrombocytopenia with the area under the curve of carboplatin that was given, while the open circles and the curve on the left are the amount of thrombocytopenia you got when you weren't giving Taxol. And there does seem to be a platelet sparing effect of the Taxol combination, and that's probably what's made that combination of carboplatin and Taxol so popular. In lung cancer, there's still a split. Carboplatin's used by some people, cisplatin used by others. There's quite a few randomized trials comparing the two, and some of them have shown carbo to be inferior, some of them have shown it to be much the same. But quite a lot of people, particularly in Europe, believe that cisplatin is a bit better than carboplatin for lung cancer. Personally, I'm not sure whether that's true or not. In testicular seminoma, we had a patient, when we were doing the phase 1, who was Maltese, therefore he had to pay for private medicine in London. And he'd run out of money, so he said, well, could he go on our trial? So we gave him a course of carboplatin, and after the first dose, he went into complete remission. And in fact, carboplatin is still used as a single agent for adjuvant therapy of seminoma now, and 2 courses are normally given. However, for testicular teratomas, other germ cell tumors, cisplatin remains the treatment of choice because there have been 2 trials that showed that carboplatin had a slightly higher relapse rate than cisplatin. And I'm perhaps risking something talking about teratoma with Larry Einhorn in the room. But one of the things about the formula is you can invert it. And so if you know how much carboplatin somebody was given, and if you know what the glomerular filtration rate was, you can work out what AUC they were exposed to. And this is a plot from a bunch of patients, 78 patients, I think, were treated with carboplatin for seminoma at the Royal Marsden Hospital. And I wasn't in charge, and they did various different things with the dose. But you can see here that all the relapsing patients had AUCs of under 4. If you split that up and do statistics on it, you find that there's no cutoff where you don't get more relapses under 4. And the predicting relapses based on the dose given on a square meter basis doesn't work. Now, the next thing about the teratoma story is that creatinine clearances estimated by creatinine-based methods tend to be lower than those you get with a 51 chromium EDTA. So if you use that, you have to give the higher, you have to increase the AUC you're aiming for a bit because you're going to get a figure that's too low. And when the Medical Research Council in England set up their carboplatin randomized study in teratoma, they asked me about this, and I said, what you should do is add 10% to the dose if the investigator is using a creatinine-based method. And what they did was they put it in the protocol telling people to reduce the dose by 10%. They just got it wrong. And if you actually look at the data and take that cutoff, I think the slightly higher relapse rate they had could be explained by that. This was a US study which was done Marsden et al. 1993, again showing a slightly higher relapse rate for carboplatin and cisplatin. But they did only give the carboplatin every 28 days. And I think most people know that teratomas, you need to keep hitting them hard. And the doses they gave, 350 to 500 milligrams per square meter, were probably too low. You'll remember my teratoma patient I showed you had to have 1,000 milligrams to get a sensible AUC. So although I don't think anybody will ever revisit using carboplatin in teratoma, in adults at least, I think if the doses have been more carefully worked out, the trials might have shown equivalent. In pediatric tumors, it's a different story. Pediatric germ cell tumors are quite rare, so nobody does randomized studies. And one study was done which showed quite poor results, but that was giving carboplatin at 400 milligrams every 6 weeks. Subsequently, a study giving 600 milligrams per square meter of carboplatin every 3 to 4 weeks in combination has shown survival as good as any other published trials. And there's really no evidence of nephrotoxicity or deafness, which are quite problematic giving cisplatin to children. And I believe in most centers, pediatric teratomas are now treated with carboplatin containing combinations. Cisplatin never really found a place in breast cancer. There were lots of other drugs that were used, and maybe women didn't like the toxicity, and the few trials that were done didn't look particularly better than other ones. But it has recently found a place in HER2-positive breast cancer, mainly due to the work of Sid Slayman in Los Angeles. And tumors where the HER2 gene is amplified used to have a worse prognosis than those where it wasn't. And the best drugs for treating were doxorubicin and paclitaxel. Then trastuzumab, which is of course an antibody against the HER2 gene, was introduced, and that further improves the survival in HER2-expressing breast cancer patients. In fact, so much that they now have a better prognosis than the ones who don't express HER2. But the problem is that trastuzumab and doxorubicin are both cardiotoxic, so that the problems with cardiac failure as a result of the therapy became a problem for treating breast cancer. So what Cisplatin did was he tried substituting carboplatin for the doxorubicin. And if you look at, you can ignore the bottom line because that didn't have the trastuzumab. If you look at the top two lines, the dotted one is the dox, and the solid one, no, I think it's the other way around. Anyway, one of them has got the carboplatin and the other has the doxorubicin. You can see they're doing very, very similarly. The difference is that on the carboplatin arm, you're not getting any acute leukemias. You're getting much fewer congestive heart failures and not really any difference in the responses. So for that particular group of patients, it looks as though carboplatin is useful for breast cancer. It's also used in some other circumstances in breast cancer. I think I'm nearly there. This is just from the Wikipedia. I've touched on some of the cancers where sort of I'm interested in. I think it's something to learn, but carboplatin is also used in a wide range of other cancers. And it's probably one of the most commonly prescribed anti-cancer drugs now. And interestingly, the breast cancer studies show us that the oncogenesis by the anti-cancer drugs themselves, which can occur with many of them, seems to be quite unusual with carboplatin, certainly less common than it is with doxorubicin. I think we'll hear a bit more about it later. We still have trouble getting the right dose. And I think there's still work to do to set up methods that makes it easy to get the right dose. If you do an isotope clearance and work very carefully, you can get the right dose, but that facility isn't available everywhere that carboplatin is being used. So I'd like to acknowledge all the many people without whom, I don't know whether I am Mr. Carboplatin or not, but without them I certainly wouldn't be. And also University College London, who have supported me for the last several years. Incidentally, a lot of the work to develop it was done by charity by the Cancer Research Campaign in the United Kingdom. So thank you very much indeed.
So thank you for that fantastic story. We have a rich and full schedule for today, and we're just getting started. So I think we're going to take our questions into the break and have our discussions with you in the break. So we have a break right now for the next 10 minutes. So we're going to start up, less than 10 now, 8 minutes. At 45 after, we're going to start up again the next session. Okay, but there's plenty of time to mingle and interact and speak with Hilary and others.
Oh, I see.